Thank you, Peter. And I'd like to thank Ali Rezai as well, and Parag Patel and the rest of the selection committee. It's a real honor to be able to present here at this meeting. The last meeting I was at was the Acapulco meeting that you heard about. And the field has really changed quite a bit. Last night I also had the pleasure of having dinner with Dr. Benabid, and we were talking about aha moments. And he was telling me the story of when he first stimulated a patient using 100 hertz stimulation, and he watched the tremor stop. And he asked the technician to stop the stimulation, and the tremor returned. And he said, okay, stimulate again. And then the tremor stopped. And you could still feel that excitement, and it was over 25 years ago. And I had a similar experience when I was a second-year medical student at Johns Hopkins. I met a retinal surgeon, Eugene Dewan, and he invited me to the operating room the next day. And I had never been in an operating room. And so as a second-year medical student, I jumped at the chance, and he told me that they were going to be doing an experiment. And this was the first experiment where they, under local anesthesia, were going to place a single wire in this patient's eye and electrically stimulate. It was a blind patient. And so here I am, a second-year medical student in the operating room, worried that I was going to contaminate the sterile field, and watching. And this surgeon put an electrode, holding it in an awake blind patient with retinitis pigmentosa, holding the electrode as close as he could to the surface of this patient's retina, while some engineers turned on the electrical current. And the patient said, "I see a spot of light." And then Gene took a second wire and put it in his eye, in the patient's eye. So they had two wires, and they turned the current on both electrodes, and the patient saw two spots of light. And at that moment, I was hooked. As with an engineer, you heard Peter talk about I had an engineering background, and I figured at that point, all we need to do is more electrodes, and we'll be able to create pixelized vision, much like lights on a scoreboard. So I was pretty naive. I don't think I appreciated how difficult that challenge was going to be, but it was clear to me at that moment that it was going to be possible. So a few weeks ago, Second Sight went public. We're listed on the NASDAQ. Appropriately, our ticker symbol is EYES. So because of that, I have to tell you that despite what my 3-year-old might believe, I can't predict the future. The patients that we're treating today with the Argus II retinal prosthesis are patients who are blind, obviously. There are about 39 million people today who are legally blind in the world, but many of those folks are blind because they have cataracts, things that we can treat today using currently available technologies. But about 8 million of those folks are currently untreatable. There's no, other than the Argus II, there's no therapy for these folks that are blind. And these 8 million people break down into 3 groups. Really 2 main buckets. One is diseases of the retina, or the outer retina, where the optic nerve is still intact. And so the 2 most common examples of that are retinitis pigmentosa and macular degeneration. And then there are about 6 million patients, though, whose optic nerve is damaged, whose the cable going from the eye to the back of the brain isn't functioning. And so today, our FDA approval is for a disease called retinitis pigmentosa. And we've just gotten approval and are going to start implanting the first subject in a couple weeks in the UK for macular degeneration. I'll go on. A few months ago, CNN did a piece on Second Sight, and I think they did a very nice job of explaining the technology. So we'll play that video and let them tell you about it.
This is CNN.
Out of our 5 senses, sight is the one that we rely on the most to experience the world around us. About 75% of the sensory neurons in our brains are processing visual information. But imagine for a moment that you are blind. Once the eyesight begins to diminish, there may be no getting it back. But now an implant, known as the bionic eye, is giving some blind people a second chance. The implant is no larger than a pencil eraser, and it is surgically placed on the retina inside the eye. The resulting vision might not be perfect, but it is life-changing.
You climb up the steps? Yeah. Okay.
For 55-year-old Roger Ponce, it began early.
I was 14 years old. I flunked a eye test in 9th grade at the school eye test they give you.
Slowly, over time, his eyesight began to fail.
By in my late 20s, I voluntarily gave up my driver's license because I was afraid of like hurting a kid or getting in a wreck or something.
Roger was diagnosed with retinitis pigmentosa. It's a hereditary eye disease that damages the retina. That's a layer of tissue in the back of the eye that converts light to nerve signals and sends them to the brain. He knew that one day the disease would leave him completely blind. But it didn't stop him from living his life or from getting the girl.
He filled my arms full of daisies. I mean, he really swept me off my feet. He courted me.
Yeah,
Roger married Terry 20 years ago, and 5 years later, he lost his eyesight completely.
I fell in love with a man, not his eyes. I don't think I cared, and I still don't care what happens to the physical part. I mean, this is my husband, this is Roger, and I don't care. Do you? Whatever happens to us, happens to us. As long as we have each other, we'll work through it.
As long as we're together.
Then one day, he heard about the Argus II. It was a combination of a camera and a pair of glasses, and an artificial retina implanted inside his eye. Together, they could restore some vision. Nearly 200 miles away from the Ponce's home is the Kellogg Eye Center. It's at the University of Michigan in Ann Arbor. Roger's been a patient here since that failed eye test in the 9th grade. It was the first call he made about the Argus II.
Retinal prostheses have been in the making for some time.
Yeah, I don't have no pain in it.
Okay,
don't hurt or nothing.
There are a number of groups around the world who have different approaches to patients with no vision. In the US, the leading sort of group was Second Sight. And they've spent 20 years making this product, and we've heard about it for the last number of years.
The doctors were intrigued. The procedure was happening in other countries like Germany and France, but it was not FDA approved as of yet. It was risky, and the rewards seemed low. But Roger didn't look at it that way. To him, any vision was better than no vision. He was a good candidate for the operation because his retina wasn't destroyed by the disease. Last year, the United States Food and Drug Administration approved the device, and on January 22, 2014, Roger Ponce became the second person in the United States to get the implant. Here's how the device works. An implant no larger than a pencil eraser is placed on the surface of the retina inside the eyeball. A pair of glasses with a small video camera mounted on it captures images and then converts them to small electrical pulses. The pulses are then transmitted wirelessly to the electrodes on the artificial retina. Any remaining cells that haven't been damaged by the eye disease are then stimulated by those pulses, leading to a perception of light patterns in the brain. The operation itself lasts around 4 hours, but the recovery takes a few weeks. In the meantime, all Roger and Terry could do was wait and hope.
It takes a few weeks for it to heal up, and then they give you the glasses probably 3 weeks to a month after the surgery. First of all, they test, and they turned it on, and they said, we want to check the computer chip in your head, make sure there's no pain. I was fine. Well, before they turned it back on, they're like, you probably won't be seeing nothing for a while, so don't really expect nothing. They turned it back on, and I'm sitting there, I'm thinking, there was light flashing on the screen, wasn't there? And they're like, yeah. And I looked up, and I said, there's a light up there. And I turned around, and the 3 lights in the room, I pointed right at them.
The surgery was a success. It appeared Roger was going to be able to regain some vision with the help of the glasses and the implant. But none of this was going to be easy. After years of not processing visual information, it's not just Roger's eyes that need to learn how to see again.
You have to go through the process kind of like learning a new language. You have to retrain your brain to see and understand those images that are seen.
So every week, the Ponces drive to Ann Arbor for rehab. 3 hours there, 3 hours back.
Yes, it's been hard, and yes, it's been expensive and a little rough. But what would you do to see again? How far would you drive? I will do whatever we need to do to make this happen.
So what I want you to do is just hold them up, and it's going to be like we're completing a puzzle. So I want you to use...
Today he was working on doing his low vision rehab. They start out in the beginning really just teaching them what they're seeing. So how do they understand exactly what they're seeing? What's the difference between the dark lights, or when they see the lights, and when they don't see the lights? How to interpret what a shadow is, what's a light, and then learn about interpreting lines and things like that.
This one is round.
Yep, so what shape is that?
Circle.
Perfect.
Throughout the process, the biggest question remains: what do patients like Roger with the bionic eye actually see? It's not perfect vision by any means. There's no color, there's no detail, there's only flashes of light. But it's enough to get around. And that's more than Roger had before.
Wait, there's your monkey book.
You gonna show me monkey book?
Show Grandpa your monkey book.
It's been pretty awesome. Yep. I can tell when my grandson runs around the house. I can tell when people step in front of me. Just, you know, every day it's something small, but it's something different.
I remember when we went to U of M, and they said he had end-stage retinitis pigmentosa, and there was absolutely nothing they could do for him. Nothing. And just to go, they didn't say go home and be blind, but that was basically it. There were no treatments,
Just
nothing.
go home and live life.
So that's another reason I think this ARGIS 2 is huge. Nobody else is going to really have to feel like we did that day. They're going to hear something different than we heard that day. They're going to hear, well, you're end-stage retinitis pigmentosa, but there's this new ARGIS 2 that's been developed. Maybe you're eligible for that. Let's take a look at it. Sounds a lot better than go home and be blind, doesn't it?
There have been 4 total operations performed at Kellogg this year. It's one of only 2 clinics in the United States currently doing the procedure. 10 more are now working to add the operation. The American Academy of Ophthalmology notes that surgeries like this do come with potential complications, and the procedure is still not available to the mass public. But they also note that for someone who has no vision, this is an outstanding breakthrough, even if the vision is still only rudimentary. And with the progress of patients like Roger, there is growing hope for the bionic eye and a second chance.
You gotta believe in yourself, and I always have, that someday, yes, I would see again. Like I said, I didn't know how, what, or when, but it's happening now.
So Roger was one of the first commercial patients, and the clinical trial that led to the FDA approval was, I'll tell you a little bit about the trial. It was conducted, it was an open label trial. It was a 30-patient study. We enrolled those patients in 10 centers in the United States and in Europe. And one of the nice things about neuromodulation in general is that the device, we can test the device, the patients with the device on and off, so the patients acted as their own control at each time point. And what you see here, we had to develop a number of tests, a number of ways of testing these patients to assess whether they could see or not. We weren't taking patients from being completely blind all the way to normal vision. We were taking them from being blind up to something that was typically considered a low level of vision, but usable vision. And so the question was that nobody had ever developed assessments for these folks because there were never any treatments available. So part of our process, even before we started the clinical trial, was engaging the low vision community and coming up with these assessments. And I'll just briefly tell you about some of them. One of the tests was a square localization test where we would ask the patient to touch a square, a visual square on the monitor. And you can see here that with the system on versus the system off, pretty much with the system off, the patients would just randomly touch the screen because they were completely blind. And with the system on, they could get with very high accuracy hit the actual box. And importantly, that performance was sustained out many years, and we have patients now out over 5 years that were implanted during this trial. We developed a number of other tests, a moving line where we asked the patients to draw on the screen the direction of motion of a moving line, and again, there were statistically significant changes, and the patients could follow that line, and those changes were sustained out to 5 years. A more difficult test was a 4 alternative force choice grading test where we actually asked patients to identify the direction of gratings that were either vertical, horizontal, up and to the left or up and to the right that were presented for only 5 seconds. Again, a number of patients could do that task. And using this approach with finer and finer gratings, we found that in the clinical trial, the best patient could detect a grading that was equivalent to 1200 level of vision. And subsequent to the trial, we've been working on software improvements and using some changes in software, we've actually gotten the patient up to 2200 on a similar test. So in addition to the visual test where we were able to show that the patients saw vision, one of the questions the FDA asked was, could they actually use that vision? Could they actually do something with the vision? So we developed a couple orientation and mobility tasks where we asked the patient to find a door at the end of the room. And again, these are, you can see on the bottom without the system or with the system turned off, the patients would essentially walk at random to the end. And with the system turned on, they would have a very high level percent success. Success was defined as touching the door with their hand at the end of the room. An even more difficult task was following a white line, where success was defined as being on the white line at the very end. So the patients were given three different lines where the line either was going to go straight or to the right and to the left. And these were, you can see, really impossible tasks with the system off. And with the system on, patients could do this quite well. The FDA also asked us to have independent low vision specialists assess these patients and write narratives about their experience, both interviewing the patients and observing them. And these are just a couple of the snippets from that test that we call the Flora. The patients described and then were observed by these low vision experts, locating doors and windows. And a funny story was one of our patients came in actually quite upset with us one day, and we asked why he was upset. And he said, well, his wife realized that he could tell the difference between light and dark laundry and now asked him to do the laundry. Other stories that we heard from the patients were being able to stay in a crosswalk. So very similar to that line task that we developed. Even with a cane, patients can cross the street, but what happens is they tend to veer into traffic because they have no way of orienting themselves, and so they feel very unsafe, particularly crossing streets. And with the Argus 2, they're able to follow the crosswalk and feel much safer. So that's something that we heard quite often. The other thing that we heard very often is a feeling of connectedness to other people. So being able to actually see the person that they're talking to and make contact and know where they are and know that the person hasn't turned away or walked away is something that we heard very commonly from patients during the trial. And of course, enjoying being visual again. And we can all imagine just by closing our eyes that the difference between having no vision at all and then getting some vision back and being part of the visual world again. And I could go on with these examples, but I think I'll share another video this was a video that two months ago, Duke University implanted one of their patients and made a video of the first day that they turned the device on. And they posted this video on YouTube, and it got over 1 million hits in the very first week. And if we can play the video, I think you'll see why.
Claire, what can you see right now? Nothing now. Nothing now. Everybody ready? Okay. So 3, 2, 1, start stimulation. Yes.
Can you see?
Oh my goodness. Wow.
Can you see, Larry?
Yes.
Oh my goodness. Can I give him a kiss?
Yeah.
Oh, yes.
Wow.
Can you really see?
Yes.
Oh
it's
my
flashing.
goodness.
Big time flashing.
They're flashing? Oh my goodness. That's so cool.
Larry Hester, 66, has been blind for half his life from a condition called Retinitis Pigmentosa. In September, an electronic stimulator was surgically implanted in his left eye. On October 1st, Duke eye surgeon Dr. Paul Hahn turned it on for the first time.
So the journey is just beginning. This is day one of hopefully the rest of his life. I think turning this device on allows him to experience a whole new world. And it's not vision as we traditionally know it, but it's a whole new type of vision for him.
Are they uglies? Is that what they are?
Yeah.
They are cool. Only the coolest from Pop.
That's right.
It was incredible. it was bright and it was significant. And I just had to take a deep breath, and I was just, it was hard to articulate what I was feeling, but I wanted to share it with everybody I could grab at the time and hug.
Thank you for this opportunity for Larry to see a little bit again.
These patients have been essentially living in blackness in most cases for many, many years. And so what this is doing is almost, if you could imagine, almost a pixelated type of vision where they are getting stimulated with flashes of light when they perceive something that's bright, and when their camera is picking up something that is darker, then they get less flashes of light.
Yeah. Oh, look, he did it. He went right to it. Woohoo! All right.
That's pretty cool.
Way to go, Larry.
Wow.
I mean, that's significant right there.
Wow, that's cool. That's great. That's amazing. Huh.
Isn't that wonderful? It's only going to get better. You know it?
Hold on, I got to do that.
So the patient essentially has to try to determine what those flashes of light mean, and in many cases it can help them determine where a window is, light coming in through the window, or where a doorway is, and essentially help them navigate the world around them.
And it's going to be challenging because you're going to feel it with your hands and all that stuff. But
It's so basic. The light is so basic and probably wouldn't have significance to anybody else, but to me it's meaning I can see light and we can go from here.
This is a milestone to me on many fronts. I think on an individual level for Mr. Hester, it's a milestone because, again, for the first time, he can experience something that he hasn't experienced for 30 years.
15 minutes of fame. I said he's going to enjoy every minute of it.
I think this represents a new generation of medicine where, rather than just treating or just watching as patients lose vision or trying to stabilize vision, for the first time, we can artificially restore vision. And I think this opens the door to a whole new generation or era of medicine, which is almost like science fiction.
So a little bit about the last couple years. I won't walk you through the 25 years that it took to develop this, but the FDA convened a panel back in 2002. They brought in 23 members. It was the largest panel that they had ever had in the ophthalmic devices division, I'm told. And there were 19 voting members, and the folks voted 19-0 in favor of the benefits outweighing the risks of the Argus 2, and that led to the FDA approval in February of 2013. And then later that year, Medicare also authorized payment for the Argus 2 beginning in October of 13. And the coverage policies are still being developed in a number of local areas. So today, Argus 2 is commercially available in 10 centers in the US. These are the current centers. We're hoping to double that number over the coming year. And it's also available in another 10 centers in Europe and the Middle East, and we're also hoping to double the number there as well. So I mentioned earlier the macular degeneration study that was recently approved by the MHRA in the UK. So we're going to be beginning a study in Manchester, and that study is listed on clinicaltrials.gov. And so the first patient will be implanted in just the next few weeks. But the thing that we're really excited about, and is I think especially potentially interesting to this audience, is the Orion 1. So our plan is to take our current stimulator and use it in the visual cortex so that we can help all 8 million patients who are blind, so including the patients who have a damaged optic nerve, and essentially bypass the optic nerve and directly stimulate the visual cortex. And this is our current concept for the device, and we plan to be in animals with this device in the coming months, and hope to be in patients within a couple of years with this stimulator. And it's essentially the same as our Argus 2 with a different electrode array. And it might sound a little far-fetched to directly stimulate the visual cortex, maybe not to this audience, but in fact, we're not the first ones to have done this. Going back as early as the 1960s, Professor Brindley in the UK actually stimulated patients' primary visual cortex, and produced phosphenes. In the 90s, there was another effort at the NIH led by Terry Hambrecht that also was able to produce phosphenes in a patient. And more recently, and with more subjects, William Dobell at the Dobell Institute implanted 16 patients in Portugal with bilateral implants, and these photographs that you see here were part of Dobell's work. And these were with a percutaneous plug, so these were hardwired systems, and you can see the rack of electronics that the patients had to wear. But the performance was quite good with these devices, but unfortunately the devices failed. And my best guess is that it was because of the engineering. These were literally built in a garage-type operation on Long Island that I had a chance to go visit before Bill died in 2005. And unfortunately, following his death, the work was discontinued. So this is just a little bit more about our implantable pulse generator. We're very excited about this implant and the potential for the implant. It's there's 60 individual programmable electrodes. It's long-term implantable and reliable. Today we have over 300 patient years of experience using this device, and of course FDA and CE mark approvals for the retinal indication. And the can is quite small, so the IPG can is only 10 millimeters by 2.5 millimeters high, and it's also labeled MR conditional. The externals are fully programmable and has a research interface, so we're beginning to think about other applications for this technology, and the device is programmable so that we can look at some of those. A little bit about the technology and why it took 25 years to develop, and over $200 million invested. There were a number of problems that I think we underappreciated when we first started the program. One of the challenges was making that small package with a high number of electrodes. So not only were we raising the state of the art, which at the time was about 22 electrodes, but we were trying to miniaturize it into about the size of an aspirin. And that turned out to be a pretty significant challenge, as I'm sure all the engineers in the audience know. Taking electronics and putting it in the body is like taking your television, throwing it in the ocean. And we added the additional constraint that we were miniaturizing it to the size of an aspirin. And then the retinal interface also turned out to be much more challenging than we thought. The retina is like one-ply wet tissue paper, so it's an incredibly delicate tissue that we, in animals, were finding that everything we attached to that surface would tear that very delicate retina over time. And so finding a stable interface that could be stable over years and an electrode array that could be delicate enough and yet robust enough turned out to be a very significant challenge. And even the material that we chose for the electrodes, it turns out platinum, which all of the other neurostimulators that are out there today use, would dissolve on these very small scales. So our electrodes are only 200 microns in diameter. And on that scale, we found platinum would dissolve over time, and the devices would go bad. And so we actually had to develop a new electrode material, which was a lower. Not only did it had a higher surface area and a much lower impedance than platinum. And of course, the interconnect. How do you connect these large number of electrodes to this hermetic can that we have, and the parameters as well. How do you stimulate effectively? We talked earlier about finding the right stimulation parameters, and it's a universal challenge. We make these devices in Los Angeles. There's a manufacturing facility that has an interesting backstory. This was the factory where Pacesetter Systems, which is today's St. Jude Medical's Cardiac Rhythm Management Division, started, and where our devices are manufactured in the location that those pacemakers were originally made. So what's next? We're beginning to look at other applications for this technology outside of vision. We're a vision company, but we're looking and having some initial discussions with folks about partnering for using this technology for areas outside of vision. And with that, we have a few minutes, so take any questions. Thank you.